Good afternoon, everyone. Welcome to our 8th episode of the live interview series, SSI Live Cosmic Conversations. My name is Nia, and as one of the science communication interns here at Cal Academy, I will be your host for today. In this series, we will interview a different member of our SSI cohort every episode. SSI stands for Summer Systematics Institute, a research experience for undergraduates, or RU, funded by the National Science Foundation. All of the interns Sophia and I will be interviewing work directly with curators or researchers on research projects on all sorts of topics, from plants to people to arachnids. Feel free to leave any questions in the chat and we will get to them at the end of the episode. Today I'll be talking to Matteo. Can you please introduce yourself?
Hi, I'm Matteo. Pronouns are he, they. I am a bio and math major. I go to Willamette University, which is in Salem, Oregon. I'm originally from Sunnyvale, California, so Bay Area. And I'm working on Arachnology this summer.
Awesome. Thank you.
Spiders.
And it's also Matteo's belated birthday.
Yeah, it was yesterday.
Happy belated birthday, Matteo.
Thank you.
Okay, so our first question is, tell us about your day.
Okay, it was crazy. So I went out in the field with my mentor, who is a postdoc student named Maddie, and we were collecting dragonfly nymphs, which are like juvenile dragonflies, in this pond in the Presidio. It was super hot today, so like that was hard work, but it was fun. We didn't find any orb weaver spiders though, which is sad.
Like, how long were you guys out there?
From like 11 to 1:30, at least.
Wow. And what are you collecting dragonfly nymphs for? Is it for your research project?
It's for Maddie's research project. What she's doing is like a project centered on like mercury pollution and how like that increases as you go up the food chain. Like if you look at mercury levels and like dragonfly nymphs versus like the spiders that prey on them, like orb weavers, like how do those differ? Is there a correlation?
So how does that relate to your research project? Like, what are you doing exactly for this internship?
So I'm working with all of the orb weavers that Maddie's collected. And what I'm doing is that we have a bunch of this like genus of orb weavers. Like they're super hard to distinguish morphologically. The easiest way is for like the male spiders is to like look at their genitalia, which like are different shapes depending on the species. You can't do that for female or juvenile spiders because they're inside still, like they haven't fully developed or they're just internal. And we can't dissect these because we're sending them out for like mercury, like I guess like testing, which is like just it's going to destroy the entire spider. So we can't like dissect it to look at it. So we are going instead to determine species, we're going to like barcode them. So take a region of DNA, amplify it, and then use that to determine what species it is.
Can you also like determine the sex from the barcode?
Oh, I mean, we know the sex based on like whether or not like it's adult and like if we can see the genitalia or not.
Oh, okay. Okay. So like what are some like the challenges that you're facing with this project right now?
We don't know why, but the first two like primers we use to try and like get DNA from this spider did not work. There's no reason they shouldn't have worked. Like it's H2LCO, which is like a universal like primer for mDNA. Is this too like jargony?
You might need to explain mDNA.
Okay. Like mitochondria. Like there's DNA in the mitochondria. There's this gene called CO1. It's a gene that most people use for barcoding animals. And barcoding was just like amplifying a sequence of DNA that every animal has and looking at the mutations in it to see like, okay, what species is it? Because it's an important gene, so mutations are conserved. So if there are a lot of mutations, that means it's a very different species from like each other.
So bringing that back to the challenges, I know you had to go explain some stuff.
Yeah. So the challenge is that like the primers, like what we use to bind to that DNA, like the first two that we tried did not work. They just like completely failed for no reason. But I did another round of PCR yesterday and today, And it worked.
Yay.
I just saw the gel. It worked finally. So I finally have DNA that I can send out for sequencing.
Awesome. And like, what do you do once you send it out to sequence and they send it back to you? What do you do with that?
I'm going to look at like it through this software called Genius and see if there are any like irregularities with like the sequence, like you know, like we use like I don't know how they do it actually. I don't understand that technology at all. But once we do that, we're going to turn it into an evolutionary tree.
Oh, that's cool. Okay, so moving on to like, you're working on a project for arachnology. So like, what got you into like spiders or like arachnids or just science in general?
Great question. I honestly did not have an interest in spiders particularly until this summer. I also thought they were like cool, but like never like my area of like focus. I've been like more of an insect guy, I guess, since starting college. And that's mostly because a lot of the like laboratory classes that have to do with ecology at my college are focused around insect ecology. So I've been working with them a lot, and I found it like really interesting. It's just like how understudied they are, just in general. So it's become an area that I want to like explore more and more. The more I do with it.
So entomology is where your heart lies.
Yeah.
So
for now.
For now, do you think you could do arachnology in the future?
Yeah, I'm enjoying this.
You're enjoying. That's cool.
Yeah.
Like, and you didn't do. Were you like interested in science before college?
Oh yeah, definitely. I knew like from like age 7 that I wanted to be like a biologist of some kind, and like I've always been more interested in like the ecology and evolutionary bio side of stuff, and like zoology stuff like that. So that's what I decided to do in college.
Cool. Okay, so our next question is, how does your personal identity inform your science?
This was the hardest question of the ones.
Do you need me to like explain it a little bit
no,
more?
I get it. I think that like as like a queer scientist, it sort of forced me to think like beyond like categories, if that makes sense. Like people talk about like the sex versus gender, right? Like gender is like a social construct, while like sex is like a biological thing, as like a way to help with like differentiate for understanding like terminology about like being queer or being LGBT. But the way that I sort of think about it, like as a queer scientist, is that like sex itself is also a construct. It's a set of characteristics that like we've decided all correspond to like one particular gender or sex or whatever. But in reality, like they can occur, like male female characteristics can like occur in the same person. Like they're not mutually exclusive. They're just like usually happen to occur with each other, but not always. So that sort of like made me think about like, well, like are other categories like species, like are those like really like, are those something that actually exists in nature, or is it just the way that we think about it to like help us understand nature? And I think that the answer is like, it's not something that actually really exists in nature. It's something to help us understand nature. Does that make sense?
That makes perfect sense. I was just thinking about how you were like working with evolutionary biology and making like cladograms in trees. Like, how do your concept of how like species are and like sex and gender, how does that like shape your work like right now?
Great question. I would say that it has led me to focus like more on like monophyletic groups as like a way of like classifying species more than like morphology or anything, because I've like learned that like morphology, like even if like you can visually tell something apart, it doesn't mean that it's like a different species. And even if two things are identical, they could be like wildly like genetically divergent. So it is like I have started to just like, I guess, ignore like morphology as a way of like looking at species. But that's not always helpful for like being like a scientist, because it's really easy to look at morphology. It's a lot harder to look at DNA.
Yeah, you can't see the animal's DNA in the field.
Yeah.
But like, so you think species should be determined solely based on like genetics and not at all based on morphology?
I think it's whatever is useful for what you're doing.
Mm.
I guess that's my answer, is that like whatever definition of species you're going to use, you should probably base it off of like what's most relevant to like what research you're doing, and not like some universal definition.
Because there's no universal definition.
Yeah, nothing's going to work for everything.
Okay. So doing all this lab work and going through college and dreaming of being a scientist as a 7-year-old, what do you think being a scientist means to you?
It's about like a passion for educating others, I think. Like, I guess like I want to help other people understand more about the world, and like so like that's like sort of my goal for for being a scientist, is like I want like the research that I generate to like help other people like understand the world around them. If that makes sense.
Mm-hmm. Do you ever see yourself maybe taking on like an education role, like a professor to teach science? Or
I've thought about it a lot. I don't know ultimately, because I don't know how good of a teacher I am, like as like a hands-on educator. Like I'm not sure that that's really my niche, but I think that science communication is like really important, and that's why like I'm stoked to work at the academy, because like I feel like the research that I do is like a lot more directly connected to like the general public than like a private laboratory somewhere.
do you see yourself doing more things like this to like communicate science, like interviews, like social media, stuff like that?
Yeah, definitely, especially like biodiversity science. Like what we learn is only useful if like it helps like policymakers and also the general public like think differently about biodiversity. Like that's like the main goal, right, with conservation science, is to change how we interact with nature.
Definitely.
And that requires education.
Yeah, I always think like doing research is really cool, but if no one knows about it, how are things going to change?
Yeah, exactly.
Perfect. Well, that wraps up our core questions. Are there any questions for us in the chat?
There are. There's also some very strange. There's someone impersonating you, Nia.
Okay.
And
Fun.
I don't know what's going on, but we'll figure that out later. Jaquez has a couple questions. Let's see. His first question is, what is the most interesting thing that you have found during your research?
Like on spiders?
I guess. Or like, have you done research previously?
Yeah, I did research on like gall wasps for like a class previously. I think the weirdest thing I found there was like, oh God, the amount of stuff that like lives inside like a gall. Like a gall is like a tumor that insects form to like incubate their eggs in. They like on plants. And like I had to dissect a bunch of them for this project, and there were just so many like random insects that just like living in there that had nothing to do with like the research. Just like I would crack it open, and like a cloud of mold would explode all over me, and like earwigs would crawl out, and I'm looking at wasps. What are you doing here? Go away.
There's a whole ecosystem in there.
Yeah, that project sucked.
Oh no. Do you like this one better?
Definitely.
Cool.
Your next question is, what would you say has been the most valuable experience in your journey to get you where you are today? In like your science journey?
Great question. I'm not sure I know how to answer that. Most valuable experience. I mean, I just said that I hated that project, but like getting a chance to do that research is like a big reason why I stayed as like a bio major, because I was considering not being a bio major after like a year of school, because I was really tired of the intro classes. I wasn't feeling it. I was like, I don't like this. And then I got to do research, and I was like, okay, there's parts of this suck. Yeah, definitely. But also like, I could see myself doing this. Like,
what were the parts that made you stay? Like, even though you hated the project?
I mean, it's just like exciting discovering something new about like whatever you're studying. Like that just feels really good and cool.
Yeah.
It's when you don't discover something new that like is like annoying.
Bums you out.
It's like, oh, like there's an issue with my experiment. Like, oh no, like.
I get it.
But when it works, I'm like, yay!
Yay.
I have data.
I mean, sometimes no data says a lot.
Yeah, it's true.
All right, so your last question from Jacquez is, what is your dream science job?
Oh God. I don't know about like a forever job, but I really want to work in like a country like Costa Rica at some point, like studying biodiversity there. I think like that's a dream goal for me.
Why Costa Rica?
I just think that it's cool. It's like one of the most biodiverse countries on the planet. So I think it'd be a really great place to study entomology specifically.
Oh yeah, there's probably a lot of bugs there.
Oh, Argentina. I'm Argentine, so like that would be a nice little like, I guess, identity meeting science moment.
Connect it back to your roots. That would be nice.
Yeah.
Is that all the questions for us in the chat?
Yes.
Okay, then. We'll move on to our comment round. Our first question. What is the weirdest or most surprising thing that you've learned in your studies? You kind of already covered it, but is there anything else?
I mean, I've like learned like a lot of weird ecology facts, but I've learned so many at this point that like none of them really stand out as like being the weirdest.
Well, pick your favorite one, or just one.
I mean, there's that fungus that mind controls ants. I think that one's still pretty like crazy to me.
That is crazy. That's kind of.
Or are those wasps that like sting insects and then that like venom or no, like they inject their eggs into them and then like the eggs take over the insect and like force them to do like stuff to like ensure the larvae's survival.
A lot of parasitism.
Yeah, it's really crazy. Parasitic ecology is just like so crazy and so gross, but also kind of cool.
Weird. My weird fact is the fig wasps. I just think that's so crazy. They go into the like figs, lose their wings, lay the eggs, the males hatch, mate with their sisters, the females fly out and then do the same thing. Be careful when you eat figs. Okay. Next question. If you could be an animal or plant for a day, what would you be and why?
For animal, I'd probably be like an orca.
Why?
I just think that they're really interesting. Like I would want to understand like what like an orca's mind is like because they're so intelligent. So like, is it anything like being like a person? Like I kind of want to know because like they talk to each other.
Yeah, don't they have like super complex like vocalizations?
Yeah, they have their own languages.
Wow, and dialects too, right?
Yeah, because like it differs from like different like between orcapods, which is just so crazy to me.
Yeah, they must. I think that they're probably almost as smart as us, but we just don't know it.
Yeah.
The people of the ocean. Okay, our next bonus question. If you could instantly learn any skill or technique in biology, what would it be?
All of like the computer programming and software stuff that you need for data analysis. Like if I could be like fluent in coding R, fluent in coding Python, know how to use like all of the different softwares I have to download instantly, that would be great. Because I think my least favorite part is like having to learn how to use all that software. It's so tedious.
Yeah, it would be nice to just know it. But it's a good thing we're doing a workshop tomorrow. And also, ChatGPT. Okay, our last question. What is your favorite SSI moment so far? Okay,
so Nia was there for this. Some of the interns went to roller skating at this place called the Church of Eight Wheels in SF. And let me tell you, roller skating is not a talent of mine. I am in fact very bad at roller skating. Probably like one of the worst ever.
I think you were getting the hang of it towards the end.
Yeah, so like the first time I tried, I think I knocked Julia over at least 4 times, and that was like, only like going like halfway around like the entire floor. But with everyone's help, I was able to like make it around, I think at least once. So that was like a yay moment for me, is being able to like roller skate with assistance.
Well, I'm sure we'll go again and you'll get to practice some more
Yeah.
if you want to. Well, that is all of our bonus questions. We'll go ahead and wrap up. Thank you, everyone, for joining us. Before we end today, we would like to encourage you to follow us on social media platforms at CalAcademy_SSI to learn about science and natural history museums along with us this summer. Tune in next Tuesday where we will be interviewing Haley on her research in herpetology. Thank you so much for watching.